Okay, so next, Richard Phillips, Dr. Richard Phillips from membranology. So again, the membrane harvesting system. So if you've seen, the algae reactors contain a lot of water, and we have to separate the algae from the water. And you can do it through various methods, but the membranology system is the option that we went for this system. So Richard will explain his company and the products that he has there. Thank you.
Thanks, Matt, and thanks, Mikey and the CCAP team for inviting me to talk about this today. Just going to start off by going back a little bit in terms of circular economy. We've done a lot of projects within the company looking at human activities, so anaerobic digestion waste, CO2 emissions from industrial facilities, and how they can impact and be used for algae production. I know Mikey talked about some other sort of uses of algae production and grow on pretty much anything, and wastewater treatments and things like that. And then you've got to process that, because most of the algae is full of water, and then we can produce some algae products. So some of the products that we've sort of fractionated from the algae, you can see on the screen there. The picture on the bottom there with the nice colours, that's from a phycoerythrin from a porphyridium sample that we actually separated out. So the pink colour you get is actually a combination of those 5 colours there. And we've done a lot of work with the phycoerythrin in terms of not only just membranes, but other processes to try and get that up to analytical grade for high value products. So some of the market, they've got food and feeds, fertilizers, cosmetics. It's a whole range of products. Other people have touched on that today, of what you can get out of algae. So where do we fit in? So we are the downstream processing. So when you've got your algae and then you've got all this bulk solution, you want to concentrate that up. Now, people use centrifuges, they're quite inefficient. So we use cross-flow filtration systems for doing this. So how that works is you've got the hollow fiber system, you feed the solution around in batch mode, it's pumped around constantly. And what happens then is you've got your sort of, let's try a nice little porphyridium, that's going through the tubes, water penetrates through the walls of the tubes, it's just small enough, and you get this clear permeate coming through the tubes, and then you concentrate up the algae within the tank, and you end up with a nice concentrated biomass, clean permeate. And because you're using sort of filtration systems, that permeate can be reused back in your process because it'll be sterile. So again, some of the sort of bits of kit we got, that's the sort of pilot system they got in SAMS. You see me demonstrating that later on. And there's some of the benefits you got there. Lower energy, much lower energy than the centrifuge, low chemicals. But you also got other options then, like I said, water recycling, desalination if you wanted to change the membrane profile and things like that. But also then downstream processing of your concentrated material that you disrupt if you're looking for protein concentration or fractionation of those different components within the material. So some examples. So this is our smallest unit. It's our benchtop harvester, which is on the stand at the back there. And we use that for typically a sort of a carboy size. You can go up to larger volumes than that, but this will concentrate a carboy in an hour and uses less than 0.1 kilowatt hours of energy. So you can see it's very energy efficient, very useful. And you can see on the left-hand side, you've got the feed material. Then you've got the clear permeate and the concentrated algae biomass at the end. Some other works then, so we did some work with Elliot here in SAMS, looking at Phycocyanin extraction and purification using, again, our benchtop systems, where we used a kind of a microfiltration type system to separate the Phycocyanin from the biomass, and then another ultrafilter then to purify and concentrate up the Phycocyanin for further work. And then another one then is EPS extraction. So we had some samples from Glasgow, and we looked at, again, doing a similar sort of process, getting the EPS away from the microalgae and then concentrating that up. So in terms of scale, we've got a load of different scale materials depending on how much we want to process. There's benchtop equipment we have, runs from sort of a couple hundred mils up to sort of maybe sort of 50 to 100 litres of material. And then we have a bunch of pilot scale equipment. These are mostly for our in-house use, but we've got systems that we've deployed across the world. The one on the left there is a small harvester. The middle one then is our development unit. So if you're looking for a specific extraction process or concentration process, we can screen 4 membranes at the same time with about 10 litres of sample. So we don't need large volumes of material. And the one on the right then is the nanofiltration reverse osmosis system. So if you're looking for sort of tight concentration or RO water recovery systems and things like that, that's what that's for. So before I finish off, I just touch on what else we do because it's of interest. I touched on the anaerobic digestion waste earlier when we looked at processing those. We've also done work with fermentation broths. So the benchtop system would work for any sort of fermentation if you're trying to concentrate up and separate out the cells from the media. And then we've got a whole range from microfiltration down to reverse osmosis and everything in between. If you're looking for specific applications for your valorization of your waste stream. So thanks for listening. As I said, I'll be down the stand on and off today, and I'll be demonstrating in the lab the membrane filtration cross flow system. Yeah.